This is part 2 of our lecture on the Introduction to Developmental Biology. I am Ma.CristinaS.DeLasLlagas and I welcome you to my video. At the end of this presentation, you should be able to discuss some of the molecular signaling processes that regulate human development. We begin this video with the basics on gene transcription and its role in developmental biology, other regulators of gene expression, induction, and organ formation and cell signaling. Recall that the single-celled fertilized zygote divided through mitosis to become a multicellular organism, and that each somatic cells that are products of mitosis would essentially contain identical genetic instructions. All somatic cells share the same genome, whether it is a skin cell or a liver cell, a kidney cell or a muscle cell. So what then makes cells different from one another? The answer to that would have something to do with gene expression and its regulation. Not all genes in the somatic cells are turned on at the same time. At different stages of development, certain genes are turned on or off in a specific tissue. The regulation of gene expression is responsible for cellular differentiation. Cells become specialized in structure and in function. Gene expression can be regulated at several levels. It can be regulated at gene transcription level, during RNA processing. mRNA may also be selectively translated, and also during post-translational modification. Let us briefly review how the chromatin that contains the genes are organized. Genes are contained in a complex of DNA and proteins called chromatin. The DNA and histone proteins are organized into repeating subunits called nucleosomes. Each nucleosome includes a core particle of supercoiled DNA wrapped around the core histone protein. The histone core complex consists of two molecules each of histone 2A, histone 2B, histone 3, and histone 4, forming an octamer. Connecting one nucleosome to another is the histone 1, or the H1, which serves as a linker. The packaging of the nucleosomes, whether it is coiled or uncoiled, will determine if the DNA is available for transcription or not. The chromatin can exist in two configurations: euchromatin or heterochromatin. Euchromatin appears lighter since it retains the stain less. It isn't coiled, making the DNA molecule and therefore the genes available for transcription. This is the part of the chromatin that is actively transcribed. The chromatin that stains more and is coiled is called heterochromatin. Due to its coiled state, it makes the DNA and therefore the genes unavailable for transcription. Heterochromatin may either be constitutive or facultative heterochromatin. When you say constitutive, the constitutive heterochromatin remains condensed all the time. It is found mostly around centromeres and the telomeres. It consists of highly repeated sequences and few genes. Facultative heterochromatin is inactivated during phases of the organism's life. It is found in one of the X chromosomes as a bar body through X chromosome inactivation. In preparation for mitosis, the chromatin supercoils to become its shortest during metaphase. However, after mitosis, the euchromatin returns to its dispersed state, while the heterochromatic portion of the chromatin remains condensed. In females, only one X chromosome is needed for normal functioning. Thus, the other X chromosome is inactivated during early embryonic phase. The inactivation of the X chromosome, whether paternal or maternal in origin, happens randomly. In some cells, the maternal X chromosome may be inactivated, while in other cells, the paternal chromosome may be inactivated. This accounts for the calico pattern in cats, where the fur color may be orange or black or a mixture of both. The gene for fur color is found in the X chromosome. If the allele inactivated is responsible for color orange, then the fur color will be black. But if the X chromosome inactivated carries the allele for black color, then the coat will appear orange. Once the X chromosome is inactivated or becomes a bar body in an embryo, all cells arising from that cell lineage will have the same inactive X chromosome. Another means to regulate gene expression would be through post-translational modification of the histone tail. Example of this would be acetylation of lysine in the histone tails. Whenever acetylation of lysine residues happen, it decreases the positive charges of the histone. This means that there will also be a decrease in affinity to the DNA. This causes the DNA to be destabilized and therefore makes the DNA available for transcription. Acetylation therefore favors transcription. Meanwhile, methylation of lysine residues in the histone tails increases the affinity for anionic molecules, making the histone tails more attracted to the DNA molecule. Therefore, it stabilizes the DNA. This means that it will not be available for transcription. Methylation also recruits heterochromatin-associated proteins, which again favors the formation of a heterochromatin structure. Other ways to modify the histone would be by means of phosphorylation, poly-ADP ribosylation, and ubiquitination. This diagram shows how methylation and acetylation affect the availability of the gene for transcription. Here, the lysine residue number 9 in histone 3 is active for transcription or is available for transcription. However, when the acetyl group here is removed by enzymes like histone deacetylases or the HDACs, the lysine residue number 9 is now available to interact with methyl group. Methylation here is possible with the help of the enzyme histone methyltransferases. Methylation of lysine residue number 9 recruits heterochromatin protein P or HP1, which somehow prevents this gene from being translated, and therefore this silences the gene. Gene transcription is also a means to regulate gene expression. When the genes are transcribed into mRNA, the messenger RNA may later be translated into protein products. DNA is transcribed from 5' to 3' direction, and the growing RNA molecule extends from a 3' to 5' direction. This diagram shows the components of the genes that reside in the DNA. It includes the exons and introns. The exons are regions that contain sequences that can be translated into proteins, while the introns are the intervening sequences that do not correspond to any protein product. The promoter region is found upstream the transcription initiation site, here, and it binds the RNA polymerase for initiation of transcription. RNA polymerase, together with transcription factors, will bind to the data box found in the promoter region. Transcription factors allow the nucleosome to unwind, making it available for transcription. Meanwhile, the enhancers also bind with transcription factors and regulate the timing of gene expression and its cell-specific location. Gene expression may further be regulated post-transcriptionally through RNA splicing. The splice site will affect the final product of translation and may even determine whether translation can take place. Notice how the pre-messenger RNA is modified through the removal of the intervening sequences called introns, and finally, a mature messenger RNA consisting of exons, the 5' cap, and the poly A tail is formed. The initial product of transcription is the pre-messenger RNA, otherwise referred to as the nuclear RNA or nRNA. It is longer than the messenger RNA due to the presence of the introns. These introns are removed or spliced out when the nRNA is processed. RNA splicing allows for the production of different protein products from a single nRNA transcript depending on what exons are spliced through a process called alternative splicing. Example: different intron combinations will give you protein 1, protein 2, found only in bones, or even protein 3. From this recent discovery, we now disprove the one gene and one protein hypothesis. Cells are able to interact with other cells. Some cells influence or effect a change in another cell through induction. The said change is possible only if a cell is competent to respond to the signal from another cell. Cell signaling may be through paracrine signaling or through juxtacrine signaling. Watch closely the video clip explaining this concept.
To make a multicellular organism, cells must be able to communicate with one another, and to do it, cells often send out tiny chemical signals that act on the receptors of other cells. Signals can be classified according to the distance between the signaling cell and the target cell. Autocrine signals are produced by a cell and go to its own receptors, so the cell sends a signal to itself. Paracrine signals are produced by a cell and go to target cells that are nearby. And endocrine signals are produced by a cell and go to target cells that are further away. Examples of these include hormones that are secreted into the bloodstream, as well as cytokines that can be released at the site of injury and act on the brain to cause a fever. Signaling molecules, or ligands, can be hydrophobic, meaning that they tend to repel water, or hydrophilic, meaning that they tend to stay in water. Hydrophobic signaling molecules can't freely float in the extracellular space, so they're brought into the target cells by carrier proteins. Hydrophobic molecules can diffuse across the cell membrane and bind to receptor proteins inside the target cell, either in the cytoplasm or in the nucleus. Most signal molecules are hydrophilic, so they can freely float in the extracellular space to reach the target cells, but are then unable to cross the cell membrane. So to pass on the signal, hydrophilic molecules bind in receptors on the cell surface. These receptors are transmembrane proteins with an extracellular end that binds to the ligand, and an intracellular end that triggers a signaling pathway inside the cell. We can think of the cell signaling pathway in three stages. The first stage is reception, which is when the target cell's receptors bind to a ligand. It's like a key fitting into a lock. Then there's transduction, which means that the receptor protein changes in some way, and that activates intracellular molecules, the second messenger. The third stage is the cell's response to the signal. Zooming into these transmembrane receptors, there are three major classes: G-protein coupled receptors, enzyme-coupled receptors, and ion channel receptors. G-protein coupled receptors are 7-pass transmembrane receptors. These are like really long proteins that have one end that sits outside the cell and binds the ligand. Then the snake-like protein dips in and out of the cell membrane 7 times, and finally ends on the inside of the cell. The end of the G-protein coupled receptor that's within the cell activates intracellular proteins called guanine nucleotide binding proteins, or G-proteins. G-proteins are made up of 3 subunits called alpha, beta, and gamma, sort of like a flower with 3 petals. The alpha and the gamma subunits are anchored to the cell membrane and keep the G-protein right next to the receptor. G-proteins bind to guanosine diphosphate, or GDP, when they're inactive. When the alpha subunit is bound to GDP, the 3 subunits stay together so the flower is closed. But when the ligand binds, the G-protein coupled receptor changes its shape, and this allows the G-protein to release GDP and bind GTP instead, activating the protein. When the alpha subunit is bound to GTP, the alpha subunit separates from the beta and gamma subunits, like one petal opening and separating from the others. When that happens, the alpha subunit is free to interact with other proteins. It stimulates some while inhibiting others. But to act on other proteins, the alpha subunit turns GTP into GDP, and when that happens, the 3 subunits come together again. The flower closes, and the G protein is turned off. Overall, there are 3 types of G proteins: GQ, GI, and GS, and each one stimulates and inhibits a different set of enzymes and molecular pathways. The GQ protein activates the enzyme phospholipase C, which is found in the cell membrane. Phospholipase C then cleaves a phospholipid called phosphatidylinositol 4,5-bisphosphate into inositol trisphosphate and diacylglycerol. Inositol trisphosphate is soluble and diffuses freely through the cytoplasm and into the endoplasmic reticulum, where it opens up calcium channels. Since the calcium concentration is higher in the endoplasmic reticulum than in the cytoplasm, calcium flows out of the endoplasmic reticulum to the cytoplasm. The increased calcium concentration in the cytoplasm changes the electrical charge of the cell and can lead to depolarization. Meanwhile, diacylglycerol remains attached to the cell membrane and binds to the enzyme protein kinase C, which also relies on calcium to fully activate. Once calcium levels in the cell go up, protein kinase C starts to activate proteins by adding phosphoryl groups to them. Next is protein GS, which stimulates the enzyme adenylyl cyclase. Activated adenylyl cyclase takes adenosine triphosphate, or ATP, and removes two phosphate molecules, transforming it into cyclic adenosine monophosphate, or CAMP. CAMP moves throughout the cytoplasm and binds to the enzyme protein kinase A. Protein kinase A has two parts, a regulatory subunit and a catalytic subunit, and CAMP specifically binds the regulatory subunit of protein kinase A. When CAMP binds, it makes the regulatory subunit dissociate from the catalytic subunit. It's kind of like pulling the pin out of a fire extinguisher, allowing it, in this case it being the catalytic subunit, to do its job. So after dissociating, the catalytic subunit of protein kinase A is free to phosphorylate target proteins that trigger a cellular response. Finally, there's the protein GI, which is also bound to adenylyl cyclase, but in this case, it inhibits it, causing negative feedback on protein GS. This is particularly important in helping to inactivate cells. Next are the enzyme-coupled receptors. They're usually single-pass transmembrane proteins, meaning that they have only one transmembrane segment, and their intracellular end has intrinsic enzyme activity. In other words, enzyme-coupled receptors have two parts. One domain is the receptor, and the other domain is an enzyme. Each domain has a separate function, like on a Swiss Army knife that's composed of both a knife and scissors. The enzymatic domain is usually a protein kinase that phosphorylates the receptor domain. Now, there are three main types of enzyme-coupled receptors, based on the amino acid at which the receptors get phosphorylated. The first group are the receptor tyrosine kinases. These are the most common enzyme-coupled receptors, and there are many subfamilies. Receptor tyrosine kinase are generally molecules that can't phosphorylate their own tyrosine side chains. When a ligand binds, two receptor chains come together and dimerize, and they cross-phosphorylate one another at multiple tyrosine residues. This triggers a conformational change that creates high-affinity binding sites for the second messengers, which can also be phosphorylated and activated, triggering the signaling pathway. Next are the tyrosine kinase-associated receptors, which work in nearly the same way as receptor tyrosine kinases, and their name even sounds almost the same. The key difference is that they have no intrinsic enzyme activity. Instead, they're associated with cytoplasmic tyrosine kinases. When the receptors bind their ligand, the cytoplasmic tyrosine kinases phosphorylate various target proteins to relay the signal. Finally, there are the receptor serine-threonine kinases, and they have a serine-threonine kinase domain on their intracellular end. There are two classes of these receptor serine-threonine kinases, type 1 and type 2, which are structurally similar. Ligand binding brings the two together so that the type 2 receptor can phosphorylate and activate the type 1 receptor, which in turn recruits and phosphorylates various target proteins to relay the signal. Finally, there are the ion channel receptors, which are generally closed. But then they open up once they bind a specific ligand. They allow ions like chloride, calcium, sodium, and potassium to passively flow down their gradient. This leads to a shift in electric charge distribution inside the cell, triggering a cellular response. Alright, as a quick recap. Autocrine signals target the same cell. Paracrine signals target nearby cells, and endocrine signals target distant cells. Hydrophobic ligands are able to diffuse across the cell membrane and bind to receptor proteins inside the target cell. Hydrophilic ligands are unable to cross the cell membrane, so they must bind to transmembrane receptors, which have an intracellular end that triggers a signaling pathway inside the target cell. There are three major transmembrane receptor classes: G protein-coupled receptors, enzyme-coupled receptors, and ion channel receptors.
Try to answer the problems to solve. What is meant by competence to respond as part of the induction process? What tissues are most often involved in induction? And give one example. You do not have to submit this, but you are going to need this later on for your summative assessment. This ends our lecture. Thank you for listening, and remember, life is beautiful.